I am very grateful to US Poultry and Egg for providing funding for us to do research related to peracetic acid exposure. It is a very unique animal when it comes to assessing what is in the air. I always like to start off by explaining why peracetic acid is useful. It is useful because from a public health standpoint, when you get to the final consumer, the peracetic acid is gone. However, there is this period of time while it is breaking down in the industrial plant itself where there is occupational exposure for the employees. And that's really what we were assessing and where we were going to take a look at it and examine it. Now, we did do one other study. We did an initial study that US Poultry funded, and then we said, you know what, there's a lot to be done, and we were giving feedback to some of the issues. But more than anything else, we noticed that there is a lot of sensors out there and ways to do the testing, and we wanted to really get a better idea what the differences were between those different sensors. So we had three main research questions that we are trying to examine with this second study that we did. The first question was, are the three commercially available direct reading PAA monitors comparable? Because one of the challenges we received feedback from the poultry plant managers was that, hey, this plant is using this sensor, and this plant is using this sensor, and the USDA inspector is using this sensor, and they're all giving different readings. What's going on? So first we wanted to see how they compare. And then we also wanted to see how they compared if you did actual lab analysis. So you used an air monitoring pump, you collected the sample through a filter and sent it off to a lab. Then we wanted to look at if there were differences in the airborne concentration evaluated when you got further away from the source. One thing we anecdotally noticed when we were doing our first study was if you were the chiller rehang guy right next to where the chickens were coming out of the chiller, your exposures were higher than if you were the chiller rehang guy that was standing 6, 7, 8 feet away. And so there seemed to be a great improvement as you got further away from the source. And that we thought would be one of the cheapest ways to fix a problem is just move the workers a couple feet away from the source. And in my experience, I always want to come up with multiple solutions. I don't want to have just one solution to any problem. And then finally, we wanted to be able to see if there was a relationship between the airborne concentrations measured in the field and then what was measured as a titration. One of the reasons why we thought that would be useful to the plant is because they take so many titrations throughout the day. So if you could find a mathematical model and just project how much was in the air, that would be fantastic. I'm going to be a little bit of a buzzkill right away and let you know that that is, in the current way that we assess it right now, not an option. And the reason is there are way too many other factors. And when we started looking in all the confounding factors, we didn't feel comfortable providing any potential recommendations in that area. So there's currently one commercially available air sampling method, and that is referred to as the HEC method. It is based off of a French study that was done several years ago, and that has been adopted by SKC. SKC has changed their sampling method and made it more readily available. So if you wanted to order the media, you would ask for the SKC method of it. I do want to add a little note. We did this sampling last year, and then in November of 2019, OSHA published a new method that they are working on, and in that new method where they published that data, there was a comment in that report that said that this method did not meet their standards. So what the report that they just published said is that they didn't meet the 75% threshold. So any of the results I give for this method are probably an underestimate by about at least 25%. So that is something that has evolved. We are always looking at the research and seeing what's newly published, and that is something I want to give as a caveat when I report these results. There are also 3 different commercially available sensors. There are new things being developed all the time, but at the time of the study, this was what was available. And so we had the PortaSense 3. This is a sensor that has a metal tube, and it pulls air through that tube, and it measures the peracetic acid in the air. It's an electrochemical sensor. We also use the Safe Side portable monitoring sensor. That one was based off of what was commercially available at the time. I do know that they are reworking the design of it currently, but that is also an electrochemical sensor. The difference between that is it's passive. So it's not pulling air through, it's just absorbing what's in the air onto its surface. And then the 4000 series compact portable analyzer, also referred to commonly as the InterScan, is unique because it doesn't measure peracetic acid. It measures acetic acid and hydrogen peroxide and projects what the peracetic acid concentration is in the air. So that is a machine that is performing a calculation. When we wanted to do the comparison, we wanted to compare the lab sampling method and all 3 sensors at the same time. We would not burden an employee to wear this because it's very cumbersome. So my colleague Hillary and I, we stood for 3 hours at a time in the poultry plants in different locations and kept all the sensors directly next to each other so that we could get accurate readings of what the exposures were. And that's just a picture of what it looks like, but I also wanted you to see it because what you'll see visually is if you go out to a poultry plant, you might see the USDA inspector pull this yellow thing out. The one comment I do want to make is these sensors all require regular maintenance. They require you to send them back to the company for calibration, and some of them have requirements that you sign up to contracts to maintain that. So these are all things that you should ask when you go to purchase a sensor, because from a long-term stability standpoint, that's important. You also want to make sure that anybody that's bringing a sensor into your facility is properly maintaining the sensor that they're going to introduce into your environment. So you would want to be asking them about their calibration documentation as well. The other caveat to that is we saw sensors being used in the field incorrectly a lot. And a lot of it was just basic user error. The tubing, the attachments were not necessarily connected correctly. So these are all important elements that may affect the results in different plants. To give you a good understanding of where we went, we always went to the drier side of the plant first, and then we sampled in the wet side of the plant. And the reason because that is the InterScan and the PortaSense, those 2 sensors have these long tubes. And in the evisceration lines of a poultry processing plant, there's a lot of humidity. And it would require a long time to dry out those tubes to make sure that there wasn't residual moisture in there affecting the peracetic acid results. So we always went to the dry area first and then went to the cooler area. The first location, we went to whole leg cut up on the further processing side. We stood next to a production line where there were 4 jets of fan spraying. The jets were in like a fan motion spraying peracetic acid water on the conveyor belt. The floors were dry and there was no chicken being processed. So this was kind of like a control. The second, in the afternoon, we went to an evisceration line. And the wonderful thing about science and air sampling as an industrial hygienist is something always goes wrong. And it did. While we were doing that, one of the women working in the plant wanted to clean the floors. There was heavy contamination, and she started spraying bleach water. And when she started spraying the bleach water, the sensors went absolutely berserk. Every single sensor reacted immediately to the presence of chlorine in the air, reading high levels of peracetic acid, which confirms that there is a large interference with all of these electrochemical sensors measuring peracetic acid, and then if chlorine is available, it measures it as peracetic acid. Very important to know if you're trying to assess quantitatively what the exposures are. The second location we went to was, we had a thigh debone line. There were 6 jets, and this one had relatively dry floors, but the unique thing about this one is it had a shroud over it, which works really well for the employees because they are very happy that they weren't being exposed to as much overspray. And then the evisceration line, we stood as close to the USDA inspector as we possibly could because we wanted to get an idea of what they were experiencing and then also how it was standing near an OLR cabinet. And then the third location, we went on the tender line in the further processing of the plant. And this was very wet, very messy. There was water everywhere. And then on the evisceration line, again, things go wrong when you're sampling. One of the machines further upstream in the processing line broke. And so the chickens were coming down the line, not having been properly eviscerated. And the women that were standing at that location of the mirror trim had to pull the eviscera out of the chickens, and then it clogged the drains. And then by the end of our sampling period, we were standing in about 2 inches of water, which is going to affect our results because it's a surface area of water that contains peracetic acid. All of these we thought as bonuses for us as monitoring because we're seeing all the things that could possibly happen in a poultry plant all in one fell swoop. And we wanted to see how the sampling methods reacted. So what we saw in essence is, this is a whisker plot, and basically what we're looking at is a comparison of the distribution, pairing the samples together of each of the different sampling methods. The blue one is the ChemDAC sampling method. It very closely aligned with the results of the HEC method. They were very, very similar. You notice that the inner scan had a pretty tight curve. The box represents the 25th to the 75th percentile, but they did have some really extreme outliers, and those were mostly when other chemicals were introduced into the environment. And then the greatest variation, which again, I want to reemphasize that the inner scan is calculating peracetic acid by estimating it based off of acetic acid and hydrogen peroxide, and the inner scan is actually measuring peracetic acid. And the inner scan is that green box, and that had a lot, it was all over the place. A lot of fluctuation, but the sensor has a rapid response time. So when you're visually reviewing the sensor, you do see changes happen very frequently. The next thing we looked at is what happened when you got further away from the source. And so with the ChemDAC, when you were very close to the source, you saw a little bit more range and exposure. And then when you got further away, you saw a much tighter estimate of exposure, and it was slightly lower than the initial one. And that makes sense that as you get a little bit further away. We measured this at 1 feet away and at 3 feet away. We initially wanted to measure at 6 feet away, but there are so many applications of peracetic acid in poultry plants now that by the time we got to 6 feet away from one source, we were within 3 feet of another source. So we were not able to statistically look at that relationship. When you look at the inner scan, you see that there is a dramatic drop from the first exposure to the next exposure when you get further away. And you do slightly see that with the HEC method as well. There is a slight decrease as you get a little bit further away. But nothing that we could say without a doubt is going to solve your problem. So I really want you to take that into consideration. So philosophically, the real problem that we have when it comes to quantifying peracetic acid is the issue of accuracy and precision. So to go back to scientific basics, if something is not accurate and not precise, it's all over the place. Now, if something is not accurate, but it is very precisely measuring an exposure, we can adjust it to be in that more accurate measure. So we may find out down the road that we could take this HEC method, if we know for a fact that it's 25% lower than what the actual exposure is, and realign it so that it is a more accurate and precise measure. But what we have now is really in the realm of not knowing which one to believe. The gold standard for measuring peracetic acid is using a impinger method. And this method is being tested and in the process of being approved by OSHA and by NIOSH. The caveat to that is an impinger is made out of glass, and the impinger is filled with a liquid solution of a chemical agent to absorb the peracetic acid, neither of which would be allowed by the USDA to enter into a food manufacturing plant. Thus lies the problem. So some of the feedback that we've been giving when we've been talking to people like at NIOSH and at OSHA is really giving them the understanding of what's allowed in a poultry plant, what's allowed in a food manufacturing plant, and how the air sampling methods have to be adjusted to correlate with that information. A couple more things before I end. You know, sensors are fickle friends. They give you immediate results, which are great. You have a complaint in your plant, you call your QA guy, your safety manager, they come out and measure it, they give you a result. But so you can address these changing conditions, but you have these interferences that can give false results. So we had certain areas where we were looking and there would be peracetic acid being applied, and then chlorine parts wash would be not far from where it was being applied, and you'd have that interference taking place. That picture right there are two of my newest hires in our group, and I had them spray various cleaning products and use the peracetic acid sensors to see if we could get them to react. And we could get them to react to 409 containing chlorine and give us measurable readings of peracetic acid. So we did our own little challenging. They also have a high cost of maintenance, and there is an absence currently of calibration standards for these sensors, which is a huge red flag for me. If you want to go to be ISO certified, if you want to have any sort of standard protocol, if you want to be defendable in court cases, you have to have calibration data. And that is critically important with any lab sampling method. And there is also, you really want to ask about all the contracts and warranties that are required. The other thing that I really want to point out is we saw a wide variation of the jet streams when peracetic acid is applied as a spray. And we also saw employees actively changing the nozzles because they were irritated. So they were like the canary in the coal mine, and they were saying, I'm irritated, I'm gonna adjust the nozzle. And now you have this conflicting thing going on where employees are self-addressing the concern, but it may be affecting the efficacy of the treatment to the product that you're delivering. So this is just one example. It's hard to see, but there are very different distributions of how that peracetic acid is landing on the chickens. The other challenge is that we have a very small margin of error. We don't have a lot of wiggle room. We're talking about parts per billion, or you know, when we are talking about 0.2 parts per million, that is not a lot if the potential proposed ideal H value is 0.55 parts per million. And then you look at a situation where the sensors are reading anywhere between those values. So in any given moment, you could have sensors reading that you're below any exposure limits and you're above the exposure limits. So we have this small margin of error when you're jumping from one to the other. So what do we want you to know? We want you to err on the side of caution. We also want you to keep the numbers in context of the time, the location, and the distance from the source. You know, if you're using a sensor, go out when things are good and take measurements and use that as your baseline so you can compare that when you get a complaint and see what the difference is between the exposure results versus just calling a number good or not good. Please remember that the chemistry is complicated and this wants to break down. So as it's actively breaking down, you're going to experience changes minute by minute in the plant. You want to listen to the employee's symptoms, keep records of the samples, and then look for changes and trends. One thing we did notice is that if a third shift sanitation didn't properly rinse the equipment and there was a residue of sanitation chemicals, when you introduced the water that contained the peracetic acid, you'd get complaints. And that must be some sort of chemical reaction taking place between those products. Our main takeaways is that we really want you to be thinking about being strategic and deliberate about any changes. So if you're going to introduce new spray applications, please make sure that you're involving the product, the inspectors, you're talking to sanitation, waste management, line workers, and QA, and really getting an idea how it impacts the entire work environment. So that is my presentation. And I thank you very much for having me here today. Thank you.